Welcome. Here's a little basic info before we start. This is a model of our nanobot. The thin cylindrical components are emitting echolocation signals, while the thicker are the recipients. A special blue cylindrical component signifies the apoptotic signal center. More importantly, the large blue circle with the line represents the injector. The importance of the cell cycle is that it increases cell population. In the cell cycle, interphase, prophase, metaphase, anaphase, telophase, and cytokinesis occur. Interphase provides the cell with DNA replication and growth. The next 4 stages are mitotic stages. The last stage completes the cell cycle with the adult cell turning into 2 daughter cells. When cancer occurs, cell cycle occurs faster and cells are created rapidly. The deoxyribonucleic acid acts as a master plan of the cell. Inside the nucleus, this master plan provides instructions for the cell in the performance of important functions such as cell division. When genetic mutations occur like the absences of specific genes, then the deoxyribonucleic acid changes can cause diseases such as cancer. If the deoxyribonucleic acid is corrupted, then the messenger ribonucleic acid will be corrupted as well. This will occur through the process of transcription, where the deoxyribonucleic acid acts as a basis to create the messenger ribonucleic acid. The final outcome would be a corrupted strand of messenger ribonucleic acid going towards the ribosomes. Similarly, if the messenger ribonucleic acid is corrupted, then proteins would become corrupted too. Because of the process of translation, the messenger ribonucleic acid would act as a basis for the transfer ribonucleic acid. Since both of these ribonucleic acids become corrupted, then amino acid chain will also become corrupted. The end product would be a dysfunctional protein. Since proteins are enzymes, the dysfunctioning enzyme would increase the severity of cancer by decreasing the immunity of the cells. It can be concluded that the major causes and promoters of lung cancer are gene mutation, dysfunctional enzymes, and inhaling carcinogens, most of the time by smoking. But for the most part, cancer is caused by really bad cell communication. Cells don't know when to stop dividing because they aren't given the signal to stop. But enough for that. Now what you have all been waiting for. Mission Impossible, Nanobots.
Hey George.
Hey Gaurav, check this out.
Hey, help us. Come here. What? Are you sick?
No, I'm not sick. I'm smoking.
Why are you smoking double cigarettes? Actually, why are you smoking 1 cigarette? It's so bad for you. No. Maybe we should take you to the doctor.
Why is there ketchup on my face?
I tried to feed you a hamburger.
Oh. Where am I? Why am I in the hospital?
Because you passed out and you're puking up blood.
Peter George Tank, you're a heavy smoker. Well, your test results are in, and I'm sorry to say, you have lung cancer.
You have lung cancer.
It's a critical condition, sir. You have only 22 hours left to live. You should have stopped smoking.
You said it's not smoking.
You don't understand.
Okay, so why do I have this problem?
Well, a cell goes through 6 phases: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. The cell cycle is normally long, but when you have cancer, it becomes shorter. Because of this, cells will divide more rapidly, creating tumors in lymph nodes, like the ones in your lungs. This creates problems because the cell loses its signal to stop dividing. That signal comes from the tumor suppressor genes inside the DNA. When you smoke, there are chemicals called carcinogens that you breathe in. Carcinogens remove the tumor suppressing genes. But there is one way I know of that we can solve your predicament.
And what is this way?
Well, let me call in my specialist.
Hi, Jordan. I'm the specialist in nanotechnology. Now we have a solution for you. Do you know what it is?
No, I don't. What is it?
Nanobots. Well, George, you're going to be inhaling this package of nanobots along with you, into you along with anesthetics. But before I do that, will you please sign the paper?
Yeah, sure. Okay, but will I be saved?
Oh yeah. Don't worry. We're sure you're going to be alive by the time we finish this procedure. Please put that on.
Okay. Why am I signing this again?
Just in case you die.
Gaurav, Dr. Gaurav, am I okay?
I'm deeply sorry, but the nanobot mission failed.
What? It failed.
Yes, they were doing well at first, targeting the lymph nodes in the lungs and the tumor in the lung too. Because you had so much cancer and tumors, the radiation there were none left, and the nanobots have biodegraded.
Well, then inject more in me.
I can't inhale more. I can't make you inhale more.
Oh.
It's just too late. You have 4 hours to live. I'm deeply sorry.
What? No.
What happened, Dr. Nagar?
Now, since George has been smoking very heavily for a long time, and now he's paying the price by having lung cancer. Because he has lung cancer, his lung cells are mutated, and because of the lack of the tumor suppressor genes, his cells are growing very rapidly, and yeah, that is causing a tumor in George's lungs.
Is there any hope of saving his life?
You might be wondering, Dr. Pi, if there's any hope in saving his life, and there is. We have placed nanobots in George's lungs, and after an hour after his arrival, to ensure that his lungs will be safe. Now, there have been devastating effects that have taken place in George's lungs, such as a great amount of DNA damage inflicted to his lung cells. This is because of the gene mutation in lung cancer. Now, because the DNA is affected, that would make the RNA affected as well, because the RNA uses a DNA as a template to create the RNA, basically. And then with the RNA affected in transcription, then in translation, amino acids would not be created and proteins would not be created, which would ultimately lead to killing George's cells. Which is very sad because there's no hope for him right now. But we don't know. We will have to wait.
And what did they contain, Doctor?
Well, Dr. Pi, for a 70 nanometer size nanobot, we actually put trace amounts of proteins, vitamins, radiation, and a special component known as the siRNA. But in the nanobot, sticking out is an antenna, so it can communicate with cells, and we make it so that it would perform echolocation to find out whether or not which type of cell is the cancer cell. And basically, these all help treat cancer effectively, all the listed material I just stated.
What are they going to do?
Like I said, we will be using an siRNA or a small interfering RNA to silence the bad genes in a DNA in the nucleus of a cell. Now, other than that, we will also be using an antenna, like I stated, for echolocation, but in addition, we'll be using it to send out apoptotic signals to the cancerous cells. Now, apoptosis is programmed cell death, so we will be technically sending out death signals to tell the cells when cancer cells that they will be dying. It's pretty neat. And in addition, actually, you can see a visual, and this might make more sense.
Okay, so now what?
The only thing we can do now is wait, Dr. Pi.
But, Doctor, I'm still alive?
Yes, of course you are. Why wouldn't you be?
Oh, I just had this terrible nightmare that I died. Well, I mean, I didn't die, but I had to die in 4 hours.
Well, there's some good news. You have been cured from cancer. So
Oh.
no more cancer for you.
Well, I feel like a new man, so
Yeah.
I'm already feeling it. Thanks.
Yeah.
See ya.
Wait. You forgot to pay the bills.